Hello, everyone. Today we have a pleasure to host Lucie Low, who is a program manager of the Tissue Chip for Drug Screening program. Hello, Lucie.
Hi, great to meet you, ladies. How are you all?
We're great, thank you.
Yes, I think we can start our questions. So the first is, what are tissue chips?
Okay, so that's a great question to kick off with because that's what we're going to be talking about today. So tissue chips are small bioengineered devices that contain human cells. They can contain animal cells too, but we're really focusing on them containing human cells so that they can model the structure and function of human organs and tissues in a way that you can look at biology and diseases and drug testing outside of the body without using animals and in a more realistic way than using, for example, two-dimensional cell cultures. So I've actually got a couple of examples that I can show you. So here is a little tissue chip that can be used to model all kinds of different tissues within the body. I'm trying to move it in the camera so you can see it better. So you can see that there are two colors on that chip, and they represent two different microfluidic channels where the cells can be seeded, and then you can pump fluids through the chip, and so you can keep the cells alive, and you can look at all kinds of different tissues on this kind of chip. And then another kind of chip I'll show you is this one. So this is a bit bigger, it's a bit more complicated, but this actually can contain three different kinds. It's hard to see on the camera, but there's three different cell chambers on this chip, and the rest of all the blue that you see is where all the fluid goes through and things like bubble traps, so that they can feed the tissues on this chip. And so the real idea of tissue chips is that we can model your tissues, your organs, your different kinds of cells in your body, and see how they respond, whether it's to diseases or to different kinds of drugs. All kinds of different things that we can look at in tissue chips in ways that are much more realistic than, like I said, than doing it in a petri dish.
Mm-hmm. Is there an unobtainable tissue impossible to recreate? If so, why?
So there are some tissues that are harder than others, for sure. But one thing that we have been focusing on really hard here in the US at the National Institutes of Health, which is where I work, is using stem cell technology so that we can recreate any kinds of cells. So stem cell technology means that you could take blood cells or skin cells from someone, and you can actually what's called differentiate them into any kind of other cell that you can imagine. So it's really exciting technology. It's quite new technology. A Nobel Prize for this was only awarded in 2012, so the technology hasn't been around for very long. But the idea is that ultimately we'd like to be able to take skin or blood from anyone and then create any kind of tissue from their body out of stem cells. Now, currently, we can do that with lots of kinds of tissues. We can create heart cells, kidney cells, neural cells, so nerve cells, brain cells, lots of other kinds of cells, liver cells. But it's still an early kind of stage for the technology, so it can still be quite tricky to create some kinds of cells. And there are definitely some kinds of cells that we can't necessarily make out of stem cells yet. So at the moment, we can also use what we call primary sources, which is when we might actually take, for example, muscle biopsies and then use the muscle cells from that to put on a tissue chip. Or you can actually buy lots of different kinds of cells from commercial sources so that you can get holds of those more difficult to make tissues. So it's kind of an ongoing field. There's lots of work going on in the area to increase the number and the kinds of cells that we can make. But ultimately, we'd like to be able to make them all from stem cells. So we'll see how that works over the next 5 or 10 years.
Okay, but what are the main applications of the tissue chips?
So tissue chips can be used for lots of different things. So first of all, we can just model tissues on them. This means that we can start understanding how the basic biology of an organ or a tissue system works. Because if you're looking at it from an anatomical perspective, you're kind of working out what things do. And if you're looking at it from a two-dimensional aspect and just looking at what single cells do, then you're not getting a very realistic picture of what's going on in the tissue. So if we can put tissues on chips, we can start understanding just how the cells themselves just work when they're healthy, okay? Now, if we start adding in things like disease modeling, if we maybe take some stem cells from someone who has some kind of disorder or disease and make tissue chips from their cells, then we can start understanding how that disease mechanism might work, whether it's genetic or whether it's some other reason that's causing these kinds of disease pathologies. We can start using genetic engineering to actually introduce genetic abnormalities into the tissues that we have that might be healthy, and then make them into a diseased state so we can model what that happens at a molecular and a cellular level. So then we can start using them for what we call disease modeling. Once you've got healthy and diseased chips, you can start using them to test different drugs or different therapies on these chips and see how these drugs work. We understand how they work generally, but some drugs we still don't really understand how they actually fully work. And so to be able to look at that in a tissue chip in a diseased state or a healthy state can be very helpful. And then once you start introducing the stem cells type technology and you start understanding that every single person is different, as we know, obviously genetically, then you can start looking at things like what we call personalized or precision medicine, where you can start modeling, for example, you ladies on a chip. Each one of you could have your cells taken and put on a chip, and then we can start seeing how different drugs or therapies might act differently for all three of you, and then start looking at what particular drug might be the best one for you. So if, heaven forbid, someone gets sick in a few years' time, we can say, oh no, they've got sick, but we can make their tissues on a chip, and we can start looking at which drug might be most beneficial and have the most effect for you guys to actually help cure that disease. So that's that's quite a few of applications of tissue chips. But one thing we're also really interested in is putting these chips together. So it's great if you have, say, if you turn this into a lung chip, you can put lung cells in here, you can mimic the stretch and relaxation forces that your cells in your lung are subject to as you breathe. But it's not much help if it's just a lung on a chip, if you've got all the other organs in your body are also contributing to what's going on in your lung. So what we're also really interested in is putting chips together so that we can create a body on a chip, so that we can put whole systems together, or we can put sequential organs one after another, so that we can see, for example, if you take a drug, how is that drug metabolized in your liver? How does the metabolites that come from that then affect your kidneys or your gut? Or does it cross your blood-brain barrier and affect your brain cells? So there's all these different chips that we can put together so that we can start to understand how a drug or how a disease is actually modeled on a whole kind of avatar, like this, linked chips together to create an avatar of you on a chip.
Yes, you say a lot about drugs. So why don't we test all the drugs on them and fund this research more?
So that's a great question. So if we had unlimited funding, I'm sure we could start pushing towards that. But as you know, there's never enough money in the world to do all the things we want to do. But it's a really good question, but it's also it's important to note that tissue chips, while they're really cool and there's lots of exciting things we can do with them, they're not the answer to everything. So we still need different kinds of models. We still need different tools that we can use to understand different diseases and test different drugs. Tissue chips, but they're quite limited because they're still only a chip. They might have multiple cell types and they might be linked to different systems, but they're still not going to be able to fully recreate everything that makes you, you, just yet. Now, in the future, we don't know how much better these systems are going to get. We want them to be getting more complex. We want them to be able to be linked together, and we want lots of different things for tissue chips to come to fruition. But right now, we see them as a way to investigate certain things, but we're still going to need other kinds of models and other kinds of systems that we can use to help understand biology and disease and test drugs in different ways.
Right. And we know that also these tissue chips are sent to the ISS, the International Space Station. Why exactly?
That's a great question. So it's not just because it's really fun and really cool, although it is, but we partnered with NASA. So I work at what we call the National Center for Advancing Translational Sciences, or NCAS, within the National Institutes of Health. And we don't we don't have the ability to send things up into space on our own. So we partnered with the International Space Station National Lab and NASA, and we said, hey guys, we've got some really cool ideas about what we could do with these tissue chips. Let's talk about this and see if you're interested. So what essentially happened is that we have seen from the research that has taken place up in space that astronauts see changes in their physiology very quickly when they go up to space. They see changes in their muscle mass, they see loss of bone density, and it happens very quickly. So when an astronaut goes to space, they can start seeing a reduction in their bone density after just a couple of weeks. Now, the reason that's really interesting for us here on Earth is because that looks very similar to some kinds of disease states and aging states down here on Earth. So osteoporosis is a disease that's associated with aging, where there is loss of the bone density in your body, but it happens over decades. It happens over a really, really long time. So we thought, if we can start seeing these changes that look really similar to osteoporosis, for example, in just 2 weeks up in space, then perhaps we can start using tissue chips to model this and use microgravity as a tool, essentially, to accelerate some of these disease or aging-like states. So that's essentially what we are doing. We partnered with NASA because we want to use the microgravity up on the space station so that we can accelerate certain kinds of disease states, certain kinds of aging states that we just can't speed up in the same way down here on Earth. And then we can start testing drugs and therapeutics on these tissue chips when they're up in space to see how those cells are then influenced by these drugs. It might be in the same way as they are down here on Earth, or it might be in different ways. And if it's in different ways, or if the disease states start occurring in a different way than they do down here on Earth, then that gives us new molecular pathways that we can go, oh, that's interesting. Perhaps that's a new way that we can prevent a disease down here on Earth. Or perhaps that's a new target that could be really useful for a kind of drug or treatment that we can give people down here on Earth if they're getting sick. And we'll have done it by doing microgravity experiments to understand what's going on with our tissues. So that's the reason why we've done it, to try and understand disease in a whole new way.
That sounds insane, but in a good way.
It is pretty insane, but it is a lot of fun.
Exactly. And what research is now being conducted at the moment on the ISS?
So from our perspective, using tissue chips, we have funded 9 projects over the course of the last few years, and they are looking at kidney stones and kidney disease. We have projects looking at how the neutrophils of the bone marrow infiltrate into the lung after a bacterial infection. Obviously, lung infections are quite a big deal in a post-pandemic world. We're looking at tissues that are engineered heart tissues. So we're looking at how the strength of beating and the frequency of beating of heart tissues is affected by microgravity, which is similar to certain cardiomyopathies here on Earth. We are looking at skeletal muscle and how that is affected by aging and by the accelerated aging-like response we see in microgravity. And so there's a number of other systems that we're looking at. And so far we have launched all of our teams. What's really unusual for our teams is they get the opportunity to send their experiments to space twice. Now, this is really unusual because most people, when they get the opportunity to do some research on the space station, it's a one-shot thing. So they have to get it right. And they can't then say, oh, this is really interesting. We found this out. Can we fly again and do some more research? No, not many people get that opportunity. So all of our teams have had the opportunity to fly their first flight, which has basically said, this system is doing this and it works. And this is seeing what we're expecting to see. And then on their next flight, they're able to put drugs into the system. So they're saying, hey, we saw this change from the first flight. We think this drug is going to affect it. So we're going to try it and then see what the outcomes and effects of it are. So all of our teams have had 2 flights, and our most recent splashdown of research was back in March, I believe, or no, it might have been earlier than that, January and February this year, where we had some engineered heart tissues that had been up on the space station for a month, and they plummeted back down to Earth in January, February time. And our next set of experiments are actually going up just next week, we hope. So this is now the end of May. So the SpaceX 22 mission is due to fly on June the 2nd or 3rd. I think it's the 3rd right now. And we're hoping to send kidney chips up to space. So this is chips that are going to be looking at the proximal tubule of the kidney, and they're going to be looking at very early formation of kidney stones, which can take potentially decades to form down here on Earth. And obviously everyone's heard of kidney stones. They can be very uncomfortable, very painful. So if we can start understanding, using microgravity, the accelerated formation of these kidney stones and how they might be damaging the proximal tubule in the very early stages, then we might be able to start looking at drugs that can help prevent the formation of them, and therefore translate that back down to Earth and help prevent the formation of kidney stones here on Earth.
Well, congratulations.
Thanks. We're hoping for a successful launch.
Exactly. We all hope it will be successful. And you've already mentioned the pandemic. We've been living in the pandemic for more than a year now. And how can tissue chips help us with combating COVID-19 symptoms, or maybe even post-COVID side effects?
Yeah, thank you, Sophie. That's another really good question. So we have lots of different kinds of tissue chips, and by now we know that COVID affects lots of different kinds of tissues, or SARS-CoV-2, we'll call it the actual virus, right? We know that it gets in through the mouth and through the lungs and through the nose, but it has lots of other effects on the heart, on the vasculature. They've seen effects on the kidney, all these other kinds of long-term effects that SARS-CoV-2 or COVID-19 as a result can have. So it's been very almost straightforward. I wouldn't say straightforward, but it's been very obvious to a number of our researchers that they should step in where they can. They have these tools that can help model disease and understand infection, for example, and start using them to help understand SARS-CoV-2. So we have a lot of our teams who are funded by us have been repurposing, shall we say, their chips to look at SARS-CoV-2 infection. So we've had teams that are looking at the effects of SARS-CoV-2 on the kidney and how it can affect the processing of the kidney. We do actually have a team that recently published in Nature Bioengineering, I think it was, just a couple of weeks ago, which was actually using a lung chip, which was looking very specifically at how SARS-CoV-2, in this case the spike protein, is able to be affected in the lung, or the effect it has in the lung, should I say. And then the team were looking at lots of different already existing and licensed drugs to show the effect they might have as antivirals, essentially, to help stop the COVID actually getting, or the SARS-CoV-2 actually getting across the barriers of the cells of the lung. So what's really interesting from that team is that they were actually able to show that there were some currently approved drugs that might be quite useful in helping stop the absorption of SARS-CoV-2 into the body, but also they showed that some drugs that had kind of been touted as potential treatments didn't do very much. So hydroxychloroquine, which became very trendy for a while as a possible treatment for COVID, doesn't show any use in the lung chip, which is what the data is now shown further on down the line. So it's shown that tissue chips are really able to help look at SARS-CoV-2 in a way that it's very difficult to do if you're looking at patients who are sick. Animals don't respond in the same way to SARS-CoV-2, so it's very difficult to model them in animals. And so when you're using tissue chips, you're actually able to look in very careful ways how specifically human tissues can respond to this very unusual virus that, you know, popped into existence and has been causing havoc for us all. So a lot of our teams have been looking at the effects that SARS-CoV-2 or the resulting cytokine storm that can sometimes arrive as a result of infection can be modeled on tissue chips, and how different drugs can actually help prevent or reverse those changes.
Great. That's a great down-to-earth example of tissue chips. And actually going back up, so to the ISS, let's get into details. Is there a way to maybe use tissue chips in order to understand what will happen with a broken bone in microgravity?
Yeah, so that's a great question. So thinking about this one, if you think about us as human organisms, we're extraordinarily complex. Every time something happens, there's very often a mechanism built into your body to help compensate for it, or to help fix it or reverse it. And that's all called homeostasis, this kind of idea of everything trying to stay at a healthy baseline. Now, when you break a bone, lots of different things happen. So you have the damage to the actual bone, and the osteoblasts and the osteoclasts, the types of cells within that bone, they're suddenly going crazy. You have the feeling of the pain because you have nociceptors in that bone that say, hey, the bone is broken, don't touch it, don't step on it, don't put any pressure on it, this is not good. And then you have all of the immune system responses that can amplify that sort of, well, they kick off the healing process, but they definitely also help amplify the pain response so that you don't go doing more damage by trying to jump around, for example, on a broken bone. Now, obviously, you're not going to be jumping around up in the microgravity environment, but you might be bouncing off the walls and doing more damage without realizing. So I think the key point for tissue chips with answering something like that is that they would be able to answer very specific questions. So, for example, if you wanted to look at the effect of the immune response on bone damage, you could do that in a tissue chip because you can model the kinds of immune responses that your body produces and actually infuse those molecules or those cytokines or those different kinds of chemicals into a tissue chip and then see how the cells react. And you can also test different drugs. You can infuse different drugs or whatever it is that you're looking at to try and reduce that immune response and then see the effect it has on those cells. So in a certain way, for example, if you had, say, astronauts on a chip, so you were able to get some skin cells or blood cells from an astronaut, and before they went on a long-term mission, you were able to put different tissues on a chip and say, heaven forbid you get sick, but just in case, let's model a broken bone. Let's model how your immune reaction, your immune response might affect your bone in this case. And then let's try and pick the best drug that would work for you if that were to happen, that might have the fewest number of side effects, or that might be the most effective in helping dampen down that immune response or helping build up the bone tissue more quickly. So I think the answer is that tissue chips can answer aspects of what it is you're looking for. But like I said, they're a little bit limited right now because they're not as complex as we are. And however many of them you put together, it's still very, very difficult to actually model what is a full human response. And one of the most difficult things that we can model, or that we can't really model, honestly, is actually the immune response, because it is so individually personalized to each one of you individually. It's based on your genetics. It's based on your lifetime exposures to allergens and to infections, and whether you've had broken bones yourself in the past. And your immune system is shaped by everything that's happened to you. So it's very, very difficult to actually model immune responses on a chip. And that is something that we're working very hard to try and push towards, because it's such a critical response to absolutely everything, certainly to a broken bone, but also to any kind of infection, any kind of disease, even any kind of treatment that you give or a drug that you take is the response that your body has is very much based on how your immune system reacts. So I would say probably we were going back to the earlier question about what's an impossible tissue to model. I'd say it isn't impossible, but I'd say the immune system is one of the really big challenges right now.
Yeah, I'm truly amazed by this technology. And you've mentioned that we could model some diseases from the beginning, which we don't necessarily understand now. And could we, for example, model how the Alzheimer's disease starts? Because we don't necessarily understand this here on Earth. We tried to send some tissues to microgravity and see how amyloids form in microgravity, and it helped us. But could we use tissue chips for this?
Potentially, in the same way that you can use organoids, which is what some of the research that you're referring to look at the formation of amyloid plaques was looking at. I think Alzheimer's and neurological disorders are very, very difficult. I'm a neuroscientist by training, so I've spent many, many years studying the brain, and it's really difficult. So neurodegenerative disorders are very, very difficult to model because, like you said, we don't fully understand how they actually work yet. But tissue chips as a way to understand some of those molecular changes that affect the neurons at very early stages of the disease, I think are absolutely critical to helping understand some of these molecular mechanisms and also to test drugs. But I think it's also going to be very important to have a range of other tools and systems that we can still use that can ask questions from a slightly different perspective so that we can get a broader understanding of what's going on. And so, you know, one thing we get asked quite often in the tissue chip world is, well, when are tissue chips going to replace animal testing? And as every ethical scientist and human knows, we want to reduce the number of animals that are used in research. We want to refine how they are used and ask very careful questions. And ideally, we want to, you know, replace their use with other tools and other models. And so that's where tissue chips can come in helpful, because they can work towards those aims of reducing the use of animals in research. But when you're talking about things like the brain, and looking at how molecular mechanisms are affected and how the brain systems talk to each other, there are ways that you could model, for example, different regions of the brain on tissue chips and kind of interconnect them. So you could kind of create, you know, different modules of the brain and put them all together. But you're never going to be able to get a fully functioning brain system in the same way that you do currently, unfortunately, with an animal and certainly with a human. But as you know, it's even more unethical to test on humans for that kind of research. So unfortunately, when you're answering these very, very difficult questions, we think tissue chips could absolutely be useful, but we think they're going to be part of a suite of other tools that are going to have to be used to help understand these really complicated and difficult questions.
Okay, so now let's imagine that we live in a world where we have unlimited fundings and unlimited, actually, access to tissue chips. Could we use them to create personalized treatments for many patients?
Absolutely. And that's such a great question, because that's picking up on what I was talking earlier on about the idea of an astronaut on a chip. So with stem cell technology coming along the way that it is, and it's really coming along in leaps and bounds, we fully expect that, and we already know that there are instances where our researchers are creating tissue chips from individuals. We have, for example, one of our researchers, she has a multi-organ tissue system. So she's looking at bone cancer, actually, and she's got liver tissues, she's got bone and obviously cancer tissues. She's got some skin tissues, she's got vasculature tissues. So the vessels, the blood vessels in your body are very specific to the actual organ that they're close to. So it's very important that you include vascular cells or organ-specific vascular cells. So our researcher that we work with, she has got all these different kinds of tissues, but what's really exciting is that they're from the skin cells of a single donor. So she's been able to create all these different kinds of tissues from one person, and then she's able to see very specifically how the cancer is, when it's introduced into the bone in this model system, she's able to see very clearly how this one person responds to that particular kind of cancer, and then how they also respond to a particular chemotherapeutic. And so cancer treatments right now are really unfortunate in some ways, because there are not many precision cancer treatments. For example, chemotherapy is really just throwing in radiation and killing off everything and hoping that it kills off the cancer, which is why it makes people feel so terrible, because it's killing off all the other cells in their body as well. So if we can start moving towards research where we can test in tissue chips exactly what kind of chemotherapeutic is most effective for the cancer, but kills off the fewest number of the other kinds of cells in the system, then you're moving towards treatments that are very specifically targeted for individuals. We can also start looking at population differences. So there are differences based on hormones. There are differences based on genetics between men and women. We can start looking at which treatments might be more useful for men or women if there's differences in the disease, the kind of disease that they have. We can look at differences between ages. So we know, for example, that children, using SARS-CoV-2 and COVID-19 as an example, children don't seem to respond to SARS-CoV-2 in the same way that the much older population does, it's much more dangerous. So we can start what we call patient stratification, which is really looking at different subgroups of people so that we can model a disease in a particular group and then see what kind of treatment is going to be most helpful for them.
Yeah, sure. And now as a humanity, we are going to Mars and to Moon. And as we know, one of the main problems is radiation. And could we send organs or whole systems out to space where it is not protected by Earth's magnetic field and atmosphere to see how it would react to radiation doses, which we are going to take during our trips to Mars or Moon?
Yeah, so I know NASA's really interested in that question because those are some of the difficulties, and those are some of the areas that they're trying to understand, exactly as you pick up on. So the short answer is, in theory, yes, absolutely. There's no reason why tissue chips couldn't be sent off to the moon. Let's put chips on the moon. Or there's no reason that they couldn't get attached to a cube satellite, like a small satellite, and get sent around into deep space for a few years and then come back and tell us all kinds of things about radiation research. In theory. Now, in practice, it's very different. So there's biological and engineering difficulties with that. So biologically, it's actually very difficult to keep lots of kinds of tissues alive for a long time in culture. So, for example, liver cells are very hardy. You can put them in a petri dish and they do great. You can put them on a tissue chip and they function really well and they do exactly what you expect them to do. But after a certain amount of time in culture, they start to kind of degrade and they start to lose their sparkle, and they're not very healthy, basically. And so one of the problems that a lot of the biology researchers face is actually keeping their tissues alive for a long time. What's interesting is that liver cells don't last very long, but neurons can last for ages in a culture system. So I know that neural organoids, for example, can live, I think, up to 18 months, which is a really long time. And so there's opportunities there for more long-lasting tissues to potentially be used in this kinds of research. But it's still early days, it's still difficult biology. And then from an engineering perspective, if you think about sending off an automated system that is going to keep the cells alive, it's going to be telling us in real time the cells are still alive, that they're producing things that we expect them to produce. If we want to test drugs, that we can get readouts of what is happening in the tissue so that we know the drugs are working. We need to keep the tissues at the right temperature. We need to keep them at the right pressure. We need to keep them with the right amount of oxygen and carbon dioxide. So you've got to have refrigeration, you've got to have incubation, you've got to have all the media that they'll need to keep them alive and healthy. You've got to have collection devices for the media at the other end, so you can see all the stuff that the cells are producing that needs to be tested when it comes back down to Earth or when it's collected. So that's a really difficult engineering question. And even just for our chips that are going to the space station right now, that's only 250 miles away. So it might be going at 17,500 miles per hour. That's kind of the tricky bit. But it's actually really difficult just to get systems that can keep our tissues alive just for the amount of time that they need to be to get from the launch pad up to space, to sit in a space station and just do their thing for a couple of weeks, and then to come back down to Earth and survive that. That in itself is very, very difficult. So when you're talking about doing long-term missions or chips on the moon, it's a whole different level of engineering challenges that have to be undertaken and overcome. But I know certainly we're very interested in seeing what we can do to help push that technology forward, and we'd love to see NASA do that kind of experiment.
Yeah, I hope we will send them soon to Gateway Station, which will be
right?
in the moon's orbit. Yeah.
Yeah, that would be awesome. And just imagine if then you could do tissue chips from the different astronauts that are going to be doing the Artemis missions and send them to Gateway for a while and see what effects that the radiation or the microgravity might have on those particular Artemis astronauts, and then work out exactly which drugs are going to be most helpful for them so that those Artemis crewed missions are going to be successful and that astronauts will be super healthy and ready and raring to go, which I'm sure they will. But I'm sure they're excited, but it helps reduce the risk to their health and keep them healthy.
Yeah, exactly. We touched a little bit on this subject. So what is the future of tissue chips? What do you think will happen in the future?
So I would love to see, I'd love to see them go up into space further away. Like you said, maybe to Gateway, maybe to the moon, maybe into deep space. You know, we'll see what happens there over the coming years. It's going to take a lot to make that happen, but I think it's doable. Down here on Earth, one of the things that we'd like to see is a broader uptake of tissue chip technology. So when we talk about tissue chips, we're like, oh, that's so cool, that's really exciting. And it is, but it takes a long time for a new technology to become adopted and to be broadly and widely understood and available. So because the technology is new, it can be quite difficult at the moment to find people who know exactly how to use tissue chips, because the chips are developed in a lab. And so we often say that sometimes you need a specific chip whisperer who is able to understand that system and make it work. So right now on Earth, what we're really working towards are what we call reproducibility and reliability efforts, where we're really making the technology much more broadly accessible, almost so it comes kind of like a Lego type system where you can plug and play, right? You could put different chips together, you can just put them in a system, plug them in, and off they go. So that's kind of where we're focusing some of our efforts here on Earth. We want to make them much more standardized so that there's less variability between different labs that might be using them. And we also want to get them into situations where they could be helpful anywhere on Earth. So this means that they wouldn't necessarily just have to be used in a lab over in Seattle, for example, but they could be used out in Africa, where there's a new kind of disease or bacteria that's doing the rounds, and then using tissue chips to be able to see, oh goodness, what kinds of antibacterials do we have? What kinds of antibiotics do we have that we could test in tissue chips, for example? Or if you think about maybe taking them onto a battlefield. So if troops are in an area where there's some unknown chemical agents doing the rounds, which, you know, heaven forbid, we hope that that would never be the case. But if you can start using tissue chips to maybe model some of these threats on a case-by-case basis when these people are actually in the area, then you can start being able to help people stay safe and healthy all over the world, regardless of their ability, their knowledge, their training, and without necessarily the need for an expensive biological lab to be there to do all the research in. So that's what we'd love to see as well.
Yeah, that would be truly amazing to see this happen. So what are your favorite tissue chip models?
So I love all the tissue chips. I manage a lot of teams, and I would never pick a favorite team. But my favorite program that we look at, so we have a number of programs, and just to give some information, we have teams that are looking at disease modeling on chips. They're looking at rare and common diseases. We have teams that are looking at modeling pain and opioid addiction and overdose and withdrawal on chips. We are looking at, or we have teams that are looking at clinical trials on a chip, or clinical trials on a chip, which is actually really where we're encouraging these teams to use their chips in the context of decision making in clinical trials. So like I talked about patient stratification and about making a number of different people on a humans on a chip, and then seeing how that correlates to clinical trial and chip data and whatnot. But my personal favorite is the Chips in Space program. It's so much fun. I get to work with phenomenal scientists and engineers. And then when you get to go to a rocket launch and see some of the research that you've helped make happen, launch up to the space station, that's really special. So that's definitely, I think, my favorite. So I don't say I have a favorite kind of tissue chip per se, but I definitely do enjoy working on the Chips in Space program, just because it's one of the most fun.
It has to be a wonderful experience to see your program just launching to space.
It's very exciting. It's very stressful the week before launch. One thing that people never tell you is how crazy it is on the ground before launch. Our researchers are down at Kennedy Space Station right now, working probably around the clock, making sure that they get all of their systems ready to be handed over to SpaceX and loaded on a Dragon rocket in a few days' time. So no one understands fully how crazy it is the week before launch. But by the time that rocket launches, you're so elated and exhausted that it's a really special moment for sure.
Yeah, that has been an amazing talk. And thank you so much for agreeing and being with us today.
You're very welcome, and thank you so much for the invite. Eliza, Ola, and Sophie, it's been a delight to talk to you.
Thank you.
Thank you. It's been amazing.
Thank you.
So bye.
Bye.
Bye.